fun.
All right, thanks again, Mike, and thank you all for coming. My name is Nicole Fischer, and tonight I will be talking about personality in ants. There we go. All right, so tonight I have the honor of kicking off this season's UA Science Cafe series here at Borderlands on the lives of insects, from individuals to ecosystems, where we will talk about the complex and fascinating lives of insects at multiple scales. So tonight we'll start at the individual level with a talk on personality in ants, and each month we'll scale up to end the series all the way up at the community level in April with a talk on integrated pest management. All right, so when I tell people that I study ants, the first question I usually get is, well, what do I do if they're in my house? And I just tell them what I would do, which is watch them do cool stuff. You can feed them, do little experiments with them, and you can film those little experiments and send the videos to friends and family. They'll really appreciate it. And of course, this isn't the answer that most people are looking for. Most of us think of ants as pests, and we want to get rid of them. And to that end, for what it's worth, this is what happens when you dump a bunch of cinnamon in front of a line of marching ants. This is in my house. And you can see the ants here, the cinnamon here, and they won't cross it. So you can use this as a natural remedy for keeping ants out of your house, at least for a little while. Eventually they did walk over the cinnamon, and it didn't help that I built this little bridge for them too. At any rate, as I was saying, we normally think of ants as pests, but tonight I encourage you to look beyond the fact that they ruin our picnics and invade our kitchens and sometimes sting us really badly. And I hope to show you that they are, in fact, really spectacular and complex creatures. For example, the ants that you see here, these are only a subset of a much larger organized society that in many ways mirrors our own. So back in the nest where they live, which is usually somewhere underground or somewhere we can't see, there can live up to hundreds of thousands or even millions of workers. And these workers are all sisters that are highly genetically related, or they're more genetically related than human sisters. And in fact, they're more akin to identical twins in humans than human siblings. And yet, despite this high genetic similarity, they express very different behavior. And this takes the form of task specialization. So they specialize in different tasks. And so the ants that you're seeing up here in your house and when you walk out on the street, these are only the sisters who have undertaken the job of getting food to bring back to the colony. And so in this whole colony, each worker has these intrinsic differences which can't totally be attributable to genetics, but that somehow predispose her to perform one task over the other, like nest defense, nest building, brood pair, and importantly, they will maintain these differences over time, giving them a sort of personality, which I simply define here as consistent differences in behavior between individuals. And this is talking about one specific, one species. And these individual personalities are so important because collectively they can give rise to a colony level division of labor, which kind of keeps the colony humming along smoothly. And so in many ways, oh, and I just want to point out that this occurs without any central control. So there's no one ant barking orders at the other ants telling them what to do. This is all based on intrinsic differences that they have. So in many ways, their societies are much like our own in the way that we specialize professionally, and collectively we all provide the services that a society needs without having to do each job ourselves. And as Mike mentioned, just like in human societies and ant societies, there are individuals that do absolutely nothing. And these are what we call the lazy workers. And this is one of the behaviors I'm most interested in studying. So my study species is the brown rock ant, Temnothorax rubotulus is its Latin name. And these ants nest in rock crevices or under rocks, hence the name. And so here is a colony that I collected. So I lifted up this rock sort of like a clam. So this is the underside of the rock, and you can see part of the colony is stuck to the bottom here, and part of it's still on the ground. And these ants nest in pine and juniper forests, and we collect ours on Mount Lemmon, so very locally, at about 7,000 to 8,000 feet in elevation. And one of the wonderful things about this study system is that they have relatively small colonies, and so they're really easy to collect and bring back to the lab. Now, if you're taking a look at this and it just looks like a bunch of schmutz on the bottom of a rock, that's because each worker is very small, about 2 to 3 millimeters in length. So I enlarged it for you a little bit here. Here we have that part of the colony that was on the top, or on the bottom of the rock. Here's what was left on the ground. And I hope you can see from these images that they will actually take debris from around their habitat and build themselves a little wall around their nest. And we think this helps them kind of close the gap between the rock and the floor because they like very snug spaces. And just for scale, this is one of the workers compared to a penny. Just to really hammer in how small they are. But, so you can see how adorable they are, I've enlarged it for you much more, but that's about as big as I can go. All right, so after we collect these colonies, we bring them back into the lab, and we house them in artificial nests, which are simply 2 glass slides with a cardboard perimeter sandwiched in between, which kind of carves them out a little chamber. I'm gonna let the train pass, hold on.
So about how many individuals?
It can be anywhere from 30 to 500, but we go for the colonies that are like 50, 60. Yeah, they're really small. And they don't nest on the ground either, so they're not fascinating.
No, I'm good.
All right. So as I was saying, we bring them back into the lab and house them in these artificial nests. So here's the nest, here's its little entrance, and we provide them with stones. So they built this little wall themselves. We just dump it in a pile over here, and they bring it over here to make their entrance a little smaller. And we provide them with water, we give them fruit flies as a source of protein, and we also give them sugar water for carbohydrates. And if we look inside the nest here, we can see the queen right there. She's the biggest one of all. And her only job is to lay the egg. So again, she's not telling the other workers what to do. And here are all the little workers. This is a relatively small colony. This is on the smaller side. They can get bigger than this. And then these little jelly bean looking things are the brood, or the babies. So what does ant personality look like in my study species? Here are some examples, which I hope you can see on this screen. So here's a nest builder. So hopefully you can see this worker right here. She's carrying a little green stone and putting it in the place she thinks is more appropriate than where she found it. And here is a nurse, actually a couple nurses. So the ant here is cleaning or feeding one of the broods, so that's what nursing looks like. And one more example here. We have foraging, which again, very small, but hopefully you can see this worker. She's inside the food dish, and she's carrying a fruit fly, which she'll then bring back to the colony. I'll play that one more time. All right, so we have some workers that specialize in those tasks, but we have some workers that will perform many tasks and just are overall highly active, and we call these generalist workers. And then on the flip side, here are those lazy workers in action, so to speak. All right, so if you take a look here, we have some foragers carrying a fruit fly down here. Here's the queen, but if you take a look at these individuals here and these along here, These are the lazy ones, and they're not moving a muscle, and they will consistently be like that over time, and we don't know why. In fact, we don't really know what creates any of this behavioral variation, despite the fact that it's so central to the biology of these social insects. And so that is the main question of my dissertation, and I try to answer this by looking at the nervous system. Basically asking, how can differences in the properties of the senses and perception shape behavior? And by understanding this, it will not only help us better understand the natural history of a really neat organism, but we can also more generally apply these principles to any self-organized system to better understand how they can emerge from individual units acting together but following different individual rules. As a biological example, we have consciousness, which is a self-organized system, which arises from the actions of hundreds of thousands of neurons working together, but that have different properties. They receive and process input differently, much like we have colony-level division of labor from the actions of many ants behaving differently. And in both cases, these independent units are working together to give rise to something that's greater than the sum of its parts. And in addition to biological networks, these social insect systems, which have been optimized over the course of evolution conveniently for our use, we can apply them to man-made networks like data transmission in computer networks and military strategy, city and traffic planning. So why study the nervous system though? And one of my theories that I work under is that, well, the way that we sense and perceive the world can influence the behavior that we express. So for example, maybe different ant workers differ in their sensitivity to the stimuli that signal them to do certain tasks. And a really helpful analogy is if you think about any past roommates you've had. I'm sure sometime in your past or present you've had an experience with a roommate or a loved one where one of you will walk past just a few dishes in the sink, and either the sight of those dishes or the smell of those dishes will be so overwhelming that that person has to wash them right away. And so as long as that person's around, the dishes will always be done. But if they were to move out, the dishes would have to pile up pretty high and get pretty smelly before the remaining high threshold roommate would do anything about it. And because we're talking about the sight of these dishes and maybe the smell of these dishes, this implies a role for sensory sensitivity differences in creating behavioral differences. And this is essentially what we think is happening in ants. So for example, brood will emit a brood pheromone when they're hungry. And so those ants that we would call nurses, those that are tending to the brood, they would be those with the lowest threshold or the highest sensitivity for brood pheromone, according to this idea. Now, these sensitivities or thresholds, they can come from either or both the central and or the peripheral nervous system, so the two main branches of the nervous system. So maybe these thresholds are regulated only centrally, for example, by levels of some important brain chemical, in which case you might have a low threshold worker or a highly sensitive worker who responds to a low level of stimulus for a task because she has really high levels of some important brain chemical. And taking it back to our dish analogy, this would be like maybe that low threshold roommate had a really stressful week at work, and so he or she is extra irritable and sensitive to annoying things, and so they just see those dishes and can't stand them and they have to wash them. But we can also have regulation at the level of the peripheral nervous system. For example, via differences in the sensory organs, in which case we might have a low threshold worker who responds to a low level of stimulus for a task because her antennae simply are bigger. And again, taking it back to our dishwashing example, this would be like the low threshold roommate. Maybe he has really good eyesight, so he can see the crud growing on the dishes, so he knows it's time to clean them. And so what we're really talking about here is the difference between being compelled to respond to a stimulus versus merely being capable of responding to a stimulus. And my overall thesis looks at both of these levels, basically asking whether sensory differences predict behavioral differences and whether these differences arise in the periphery and or the central nervous system. which will tell us something about where in the nervous system personality comes from. And if the answer to that is yes, we might expect something like lazy ants. They might be less sensitive or have a higher threshold, in other words, to task-related stimuli, which will be reflected in either or both the sensory organs, which would represent the peripheral nervous system, and or the brain, which would be the central. So to study this, first I go out and I collect my ants. And as I mentioned, we go collecting on Mount Lemmon at an altitude of about 7,000 to 8,000 feet right near Rose Lake Canyon. And this is what their habitat looks like. So you can see the pine trees, and hopefully you can see all these rocks, and this is where they live. So we spend the day just picking up rocks and hoping for the best. Luckily, they're plentiful, so we're usually lucky. At any rate, once we bring them back to the lab, we start tracking each worker's behavior so that we can record each worker's personality. And unfortunately, they don't wear little hats and belts like this to help us distinguish one from the next. So instead, we have to paint them. And we do this using toy race car paint. And so we take each ant and secure it to a makeup sponge, and then using a needle or a straightened out staple, we dot them with 4 dots of color on their head, their thorax, and 2 on their butt. And so once we do that, we put them all back in the colony, and then we just record the colony doing their normal thing and analyze those videos so then we can tell what each ant does, what their personality is. So now we have the ants in the lab, we know how each ant behaves, and next I test thresholds or sensitivity to a variety of stimuli, for example, sugar. So this is one of several sensitivity assays that we do. Here we line the ants up, and using a little copper wire, we keep their head propped up. You can kind of see it right here. This is the ant's head, her antennae, and her body's kind of out of focus. At any rate, this keeps her antennae accessible. And then using a Q-tip, we touch the antenna with different concentrations of sugar water. And a positive response that indicates that she detected that concentration is indicated by her extending her mouth parts. So there's that little copper wire around her neck, and right about now, I touch her antenna. I know, it's a cruel trick. All right, so once we have a measure of their sensitivity, next we examine the peripheral nervous system, or the sensory organs. And we do this by looking specifically at the antennae, because these are the main sensory appendage of the ant. And so we do this by counting the number of sensory hairs, or sensilla, that cover the surface of the antenna. So just to orient you here, this picture here is an electron micrograph, a scanning electron micrograph of an ant head in profile. So you can see the ant head here, here's the eye, here are its external mandibles, and here is its elbow, its antenna, and it's elbowed. And the sensilla is the basic peripheral sensory unit. Each one houses several neurons that detect things like odor molecules and pheromones, carbon dioxide and temperature, and all kinds of other environmental parameters. So this seemed like a good place to look for variation. And in addition to looking at overall quantity, we also looked at the number of different functional types of sensilla. As I mentioned, they do have different functions, which you can see by the artificially colored image here. This is a magnified image of the first antennal segment, which corresponds to this area right here. And so each of these sensilla has a different function. For example, the orange ones, they tend to be chemosensitive, so they're a little bit like our taste buds, whereas these purple ones, the thin ones, these are mechanoreceptors, so they sense motion. And the idea behind this is that maybe certain kinds of ants, like maybe nurse ants, maybe they have more sensilla that are dedicated to detecting carbon dioxide and temperature, because they're in the nest and they have to monitor these conditions closely and keep the brood safe, while a forager might have more of those orange sensilla that are chemosensitive. Or odor-sensitive sensilla, because she's out in the world and she's looking at things and trying to find food for the colony. All right, so that's the peripheral nervous system. Finally, we look at the central nervous system, and this, of course, is the brain. And that's what the brain looks like in the head of an ant. And believe it or not, we actually dissect out the brains of these ants So here is an ant head, unfortunately decapitated, but it's embedded in wax to keep it stable. And this is hard to see, but what we essentially do is cut a window in the top of the head like that, exposing the brain and other glands and stuff, which is just this white, mushy stuff, which doesn't look so different from the wax itself. At any rate, take my word for it, the brain's in there, and then we extract it, we dye it, and then throw it under the microscope, and we can look at a few things. One thing we look at is the relative volumes of regions of the brain that are associated with sensory input and processing, which I colored on one side of this brain here in red and yellow. So the antennal lobes down here, these are similar to our olfactory bulbs. They're getting input from the antenna. And then up here are the mushroom bodies, and I colored the red side yellow, that's artificial. And these are similar or analogous to our hippocampus/cerebral cortex. So that's where the sensory info is being processed. And the idea behind this is that maybe certain workers, like lazy workers, maybe they have less neural tissue in these important sensory input and processing centers, and therefore they aren't able to process as much sensory information. And a second approach that we use is to look at the cellular level to see what the synapses are doing. So the synapses are how neurons essentially talk to each other. And theoretically, the more synapses you have, the better connected your brain is, or the more efficiently you can process new information and retrieve stored information. And so right here is a pneumothorax brain that's been stained with an antibody against a protein called synapsin, which is a protein that's found in the synapses of neurons. And so wherever there is a synapse, it will light up. And if we zoom in on the mushroom body, that processing center area here, I hope you can see the individual little balls. Each of those is a synapse. And so in that way, we can actually compare synaptic densities between different types of workers. All right, so now I want to bring this all together for you, just in terms of looking at the active or generalist workers versus the lazy workers, and tell you some of what we found. And I'm just going to breeze through this. So we know that ants vary in how active and lazy we are, so we have variation there. And when we look at sensitivity, we find that there is variation in sensitivity to stimuli, so check. And in some cases, more active ants are more sensitive to the stimuli. We've tested, for example, sugar. So that relationship is pretty solid. When we look at the peripheral nervous system, so again, this is counting those numbers of sensilla, we actually find a surprising degree of variation in sensilla number among workers, and it looks like this variation is primarily driven by chemosensitive sensilla, which is pretty interesting. However, there appears to be no relationship with behavioral variation, so whether you're active or lazy, nor is, of course, is there any relationship with sensitivity. So that's a mystery that I am still trying to figure out. At any rate, in very preliminary results, we find that there are differences in synaptic density in the brain, and whether these differences have any relation to behavior is being determined as we speak. And finally, when we look at brain volume, we see no differences among workers, so that's sort of a dead end right now. And to put this into words very simply, we find, again, that behavior and sensitivity are related, and sensilla vary, but variation does not predict behavior or sensitivity. And brain volume does not vary, but synaptic density does, and how this relates to behavior is still to be determined. So the bottom line is that there is much of this interesting story left to answer. So to close, I just want to say that the next time you see ants crawling in your kitchen, before you go and get the mouse traps, just take a moment to appreciate the fact that they aren't just little mindless automatons out to ruin your kitchen and eat your food, but in fact they're quite complex and they have personalities just like you and me. So with that, I would like to thank the Dornhaus lab, which is my lab, the Gronenberg lab, and the Rossler lab in Würzburg, Germany, who taught me how to stain synapses, and Ben Leitner for his love and support. All of the wonderful undergraduates that work with me and help me so much, and then my dissertation committee, which is going to hopefully give me my PhD someday. And thank you. I can take questions if you have any. Mike?
Well, I had a question which is totally biased, and there's no
Yeah.
reason that you would know anything about this. But in people and in rodents, they've shown that the presence of certain bacteria in the guts of the animals and in humans can actually make them more resistant to stress stimuli. And I'm wondering if you guys or your lab or anyone is looking at the role of the bacterial microbiota and how that affects animal behavior.
That's a really good question. So the question was, there's a kind of a growing area of research on microbiomes and the effects, the unforeseen effects that it has on our physiology. And Mike was asking, do we know anything about how these microbiomes, say in our guts or our gut flora, how they might influence behavior? The answer is no, no one's looking at that, and I think it's really interesting, at least at our lab. And I don't know off the top of my head of any other lab that's looking at it, but it's a very interesting question. Yeah.
I guess related to that, do you have any causal links to why different sisters in the colony have different structural and different behavioral properties? If they're all genetically related, in fact, if they're identical twins, essentially, if I understand the breeding habits right, why are they different?
So the question is, if they're so genetically similar and basically like identical twins, how do they become different? One of the major ways is through nutrition. So when they're little larvae, they get fed differently. The proportions of carbohydrates to protein will differ. And this kind of sets off different developmental cascades that then could have impacts on, the way their brain is formed, how many sensilla they end up growing, and their size, and all kinds of stuff. There is some evidence that temperature also plays a role. So sometimes you'll see, not so much in these ants, but in ants that have these really huge nests that with many chambers, the ants are constantly moving the brood from like one chamber to the next to keep them at an optimal temperature. And so if they know, for example, maybe they just had a huge decrease in their forager workforce because an animal went and ate them, they can kind of plan ahead and incubate them at the right temperature to give rise to more foragers, theoretically.
Have you looked at the epigenetics of the individual?
That is a great question. Have I looked at the epigenetics? I have not. And a really cool paper just came out on January 1st about casts and the ant casts and how they can, how their epigenome. So that's basically changes to the, how do I say this? So it's basically their DNA sequence stays the same, but changes in the way that the genes are expressed. That's what the epigenetics studies. And so in bees, we do know that nurses and foragers do have different methylomes, which is basically different ways that their DNA is methylated, which is a form of, which is one of the mechanisms of epigenetics. So when a nurse goes to become a forager, her DNA methylation patterns will change. And then if you force that forager to revert back to a nurse because you remove all the nurses, that methylome will actually reverse. And this paper that just came out shows something similar in ants where they actually used chemicals to change the methylation patterns and were able to manipulate the behaviors of the ants. Great question.
I was wondering if the foraging ants, the lazy ones, if they're stimulated to become non-lazy, remove all of the active forager ants.
So the question is, for the lazy ants, if you remove the active workers, will the lazy workers become active? And the answer is yes. So with this whole threshold idea, if all of the active workers were gone, all the tasks, the stimuli for them would just kind of add up and end up going through the roof, which would get the attention of the lazy workers. And so they'd start to do it. And there's also some evidence that as time goes by, the more you do something, the lower your threshold becomes in ants. There's some evidence for that. So eventually, if it kept being reinforced, they would become the new low threshold workers.
Is that why even lazy ants exist? Like just to help, lose all the active ants, that there's this other network of ants available.
So yeah.
What's their purpose?
So what's the purpose of lazy ants? One of the main theories is that they're a reserve force. If making new worker ants is pretty easy to do, it's not very costly, then what's the harm of like making a bunch, and if the catastrophe happens and half your colony gets wiped out, there you go. So that's kind of the predominant theory, but there's actually a student in my lab whose dissertation basically looks at that, and he looks at all these different hypotheses, like are they selfish? And so he looks at ovary development to see like are they just hanging out in the corner and like trying to lay their own eggs? And but yeah, reserve force seems to be the working theory.
I walked in a little late, you might have said this earlier, but you were using the term she when you referred to these worker ants. Are they all genetically female?
So the question is, are all the ants female? All the workers are female. And the ladies in the room will appreciate this fact. The males are just made for reproduction. They just go out, they're only made during the reproductive season, they go out, they mate, and then they die. And in fact, if you invade a nest when the males are made, so like in springtime when they're getting ready for reproductive season, you'll see the female ants are like scurrying around, getting all the brood away, trying to keep the colony safe because you've just disturbed it, and the male ants will just be like standing there doing absolutely nothing. Presumably they just don't have the sensory equipment to even realize anything's going wrong because they have one purpose only, and their sensory equipment is all geared towards female sex pheromone. So yeah, so they're all female.
Highly conserved evolutionary theory.
Sure.
Really simplistic question. How the heck do you count the sensilla?
I love simplistic questions. So I count the sensilla. Actually, this was not so simple. This took me like a year to figure out, believe it or not. What we ended up doing is we clear the cuticle. So the cuticle, which is the exoskeleton, is brown. And we can use something called methyl salicylate, which changes the refractive index of the cuticle so light can penetrate it. It changes it to that of glass, so light can penetrate it. So we can put it under a microscope after we've incubated it in the methyl salicylate, and then we can use the microscope and look at each focal plane and count the number of pores.
So you don't actually count the whole pores rather than the
Right. Now if we want something more detailed, which is something I'm in the middle of doing now, because what the sensilla actually do might matter, not just total number. To get this kind of detail, we need a scanning electron microscope. So it's a little more work and a little more money. That
So how does the queen mate? And specifically, is it like bees? Does she mate with one or many males? And does it vary between species?
It does. Okay, so how do the females mate was the question. It does definitely vary among species. So the kind of simplest answer is that the female goes out during the mating season, and she has wings at that point. The males have wings, and they all meet up and fly around, and she usually mates with one male. And then we'll go back, start her colony, dig a little hole and start her colony. However, we see in many different species of bees and ants, which are both types of social insects, that the queen will actually kind of hedge her bets and mate with many males. So and she can actually, we don't know how, but she has some control over the sperm that she uses to fertilize the eggs that she's planning to lay. And she'll store that for the rest of her life because she's not planning to leave the nest. So in some cases, yes, there are multiple fathers, which does decrease the genetic similarity. And in some cases, there are multiple queens in a colony. Yeah?
Do you have a sense of the diversity of ant species that are at that elevation?
At this elevation?
Yeah.
That's a good question. I actually don't know. Just anecdotally, there seem to be like 3 reoccurring species just from lifting up rocks. And so this is just the ants that you can find by lifting up rocks. It's, I think that, I honestly don't know how to answer that question well, but what we tend to find up there are the temnothorax ants, We find some carpenter ants, and then we find some myrmica ants, very small black ones.
And is the queen like per rock, or is it this bigger territory?
So is one queen per rock or per nest, or is it a bigger territory? We actually don't know. In Temnothorax, or the brown rock ant specifically, we have the suspicion that maybe they are what's called polydomous, which means they have many sites, sort of. And because their colonies are so small, and they nest in preformed cavities, basically, so they can't really build out their nest to suit their size. So it's possible that there are more than one site for the nest, and there are some instances in which there are more than one queen, even within one rock. But those harvester ants that we see all the time down here, with the big heads and they're kind of orangey brown, those, you'll notice, they have one hole and they're really busy, but if you look around carefully enough, you'll see there's like 5 other entrances to their nest, and it just spans like a huge way, but that's because they can build their own nest, because they're just digging out the mud.
Just curiosity, do termites behave the same way as all the ants?
So do termites behave the same way? They are social insects. They are a little different because they actually have a queen and a king. So males do serve some purpose there, but it's but it's the same idea with that division of labor. Yeah. Anything else? Sure.
I'm not quite sure how to formulate the question, but is there anything that you can extrapolate from the social organization of this ant species that you're working on and other organized systems, whether it's neurons or other social insects or more distributed organisms or anything else?
So the question is, can we apply what we know about social insect systems to other systems? And yes, we can, and that's an active area of research. I often see articles where people from the math department and engineering department, pops up on my Google alert because they use ant colony kind of algorithms as a model. So yes, I don't do that myself, but it's definitely used as a model for any kind of system that is self-organized, and decentralized. So no one main control. One more question? Sure.
I have one more question about you. What got you involved in studying entomology, insects? What's your background?
So my background is actually in cellular biology as an undergraduate, and I just, I've always loved animals, and the cell biology lab was the only place that would have me, and I learned a lot of great stuff there, but I kind of always knew that even though I had no practical background in it, that this is what I wanted to study. So coming into grad school, this was all pretty new. The way I found out that I loved this area so much is I took an invertebrate zoology class, and I learned just how crazy and weird invertebrates are, and I just fell in love with them, so I wanted to study them. All right, thank you so much, everyone.